Good evening, everybody. I'm Julie Passarelli. I'm the Exhibits and Collections Curator here at Cabrillo Marine Aquarium. And it's my pleasure and honor to introduce tonight's speaker, Dr. Christy Forsgren. But before I do that, I realized on my drive into work this morning that I wasn't sure if you guys all knew how we choose our speakers. It's mostly, most of the speakers are some way affiliated with AltaC in some way, and it's typically through SCMI. So we go through all the different faculty that are part of SCMI because they're one of the main tenets of AltaC, and so we are trying to bridge that connection. At times we do go outside the box, and one of our the last speaker for this year is from Stanford, but just so you know, that's kind of the trend. It's the Cal State system, UCLA, Occidental, and USC. So tonight we're gonna hear about some fish sex. Dr. Forsgren is an assistant professor at California State University, Fullerton. She received her Bachelor of Science in Marine Biology and Master of Science in Biology at California State University, Long Beach, and her doctorate from the School of Aquatic and Fisheries Sciences at University of Washington in Fish Physiology and Endocrinology. Christy did a postdoc at the University of California, Riverside, and was a Delta Science Fellow and researched the effect of an insecticide on the reproductive health of steelhead. Her research is currently focused on understanding the reproductive biology of a variety of fishes, including rockfishes, surf perches, and deep water fishes. She collaborates with several biologists in Southern California, including the Ocean Monitoring Program at the Orange County Sanitation District, and also serves on the Board of Directors of the Southern California Academy of Sciences. The title of tonight's talk is Ovaries, Testes, and Copulatory Structures. Oh my! The importance of understanding reproduction in fishes. Please help me welcome Dr. Christy Forsgren.
Thank you, Julie, and I'd like to thank the Cabrillo Marine Aquarium and AltaC for inviting me here tonight to be a part of the Discovery Series lecture. I'm really excited to come back to the Cabrillo Marine Aquarium. I've seen many lectures up here, and I've admired the research, and it's really an honor to be a part of that. I'm also really excited to have been invited by AltaC. I look forward to when AltaC opens, and I already have envisioned several experiments that we can do with fish pornography down there, if I'm allowed to. And also I'd like to thank all of you for taking time out of your Friday night, again, to come and look at some fish porn. This gives me hope that there's lots of closeted people out there who are really interested about how animals do it. And that's okay. I am not going to judge you a lot, because I'm sure by the time the evening is over, you will have judged me severely about what I do. And it's really amazing. I've been able to make a career out of studying fish sex. And hopefully I'll make it applicable to your daily lives by the very end of it. And maybe you'll be impressed by our gonads and copulatory structures. Maybe. You might just think I'm kind of strange. So I just want to kind of introduce fishes. I'm sure a lot of you have wandered through the aquarium and have seen the different body shapes of fishes. Some are streamlined, some are bottom feeders, some are kind of chunky. We don't judge them by their body shape, right? But they're really diverse. Our planet is covered by 71% water. And what's really fascinating is where there's water, there are fishes. And so we find a huge diversity in the fishes. This can be anything from freshwater, almost distilled water in the Amazon, to highly saline pools in the desert. So it's quite incredible that fish have adapted a physiology that can withstand all of these different salinities. In addition, fish can live in cold waters in the Arctic and the Antarctic, nearly 0 degrees Celsius, which has resulted in some very amazing adaptations where some fish don't even have red blood cells to carry oxygen any longer. We see that there's a variety of feeding modes, fish that feed on dead and decaying animals, algae, plant materials. So there's this amazing diversity of fishes out there. However, I tend to think that reproduction is really the most diverse and interesting aspect of fishes. And we see so many incredible different types of reproduction. And this is really important in understanding our fish populations, and my personal belief is that it's often very overlooked part of physiology when we study fishes. It's really important, and we have some really strange ways that fish reproduce. And I often tell my students, if you can imagine it, and I know some of you probably have some kind of crazy ideas out there, fish do it. So we see everything from internal fertilization, which we're intimately familiar with. So that's fantastic. That's really a big interest of what we'll talk about tonight. We have external fertilization. We also have situations like our local California sheephead, and it's a hermaphrodite. And this little sweet fish turns from a female to a male. So that's pretty incredible that we have this sequential hermaphrodism. We also see hermaphrodism where we have males first that turn to females. So the sex is within the same individual versus some of our other fishes that are born either male or female. In our deep sea anglerfish here, and we also have one on display at the aquarium, and I was told by somebody tonight that they think that she's kind of a hussy. I won't name who that is, Ed. But these guys are really cool because, well, these girls are really cool. When the males are small and larvae, they actually sniff out a female. When they find a female, they bite onto her and attach to her, and they become a parasite ultimately. So I know many people in the audience can relate to a parasitic significant other. These little fish are truly living that dream. Okay, so really interesting when we think about the diversity of fish reproduction. Try and advance that. Okay, so you're probably dying to know, well, how are fish babies made? Okay, the same way that we were made. Okay, egg and sperm romantically unite on this sunlit evening, and it results in this beautiful, wonderful child that will become a nuisance for the rest of its life. Okay, I'm currently experiencing a 3-year-old, so I know how this works. But what happens is they're reproducing sexually just like we do, and like the majority of organisms on our planet. So egg and sperm are going to come together to create the next generation of individuals. So here we have a picture of an egg and a sperm attaching onto it. And what we see in reproduction is it's incredibly amazing because even though we have this wild diversity of fishes and fish reproduction, the way that they physically reproduce is the same among all vertebrates. And a lot of our invertebrate species are doing the same thing, producing an egg and a sperm that are coming together. Fish, although they have to have something unique, they don't want to be like everybody else. I understand that. So they have what's called a micropyle. And a micropyle is this little hole in the fish egg. So with reproduction, you have the egg, and the sperm have to come on top of it, and they have to burrow themselves through the membrane to fertilize the egg. In the case of our fish, those fish sperm are given the extra challenge of finding the right hole to go into. So that's the one unique characteristic that we see with the fish eggs and reproduction here. So continuing on with how are fish babies made, you knew we were going to talk about testes and ovaries, right? It's one of my favorite topics. So in males, the testes are producing the sperm. So just to give you an idea of what testes look like, in case you haven't examined them on your own, here's some blue rockfish. And this is a microscope slide of a cross-section of some of those testes. And this testis is from our black perch. This is largely what we do in our lab. We take tissues, we section it with a microtome, we put it on a microscope slide, and we examine it. So you might be having some horrific thoughts back to high school or maybe your beginning biology course in college where you had to painstakingly look at microscope slides. We produce those in the hundreds in my lab. So this is what we do is make these slides. And here is what you can see is there's these little pockets within the histology. And this are the seminiferous tubules, which is where the sperm are developing. And so if we were to take the testis and separate them out, this is where we start to see the development of the sperm. And there's several different stages. So these different colors, they take up the stain in different way. And so we can actually look at the developmental progression of sperm. So it's going to take a period of time from them to go to their stem cell spermatogonia and develop into the mature sperm that's capable of fertilizing an egg. And this pattern is very well conserved. We see a similar thing in females. So females have an ovary, and here's a blue rockfish ovary here. And this is a cross-section of a zebrafish ovary. And you can see different stages of eggs or oocytes that are developing. So again, in the ovary, we have this progressive developmental stage of oocyte or egg development. One thing that's really unique in the fishes is when we see mature oocytes, or these eggs that are maturing and getting ready for ovulation, these little white granules are lipids and other nutrients that will supply all the developing embryo and early larva with all of the nutrients that it needs until it's able to feed on its own. And there's a lot of variation with this, depending on if the animal is an external fertilizer or an internal fertilizer. So there's two main reproductive strategies: that spawning, external fertilization, where you come together with a sexy mate or a few hundred of them, and you all release your egg and sperm into the environment. And you're really just relying upon chance for all of these eggs and sperm to come together and meet and create the next generation. There's millions and millions of these sperm and eggs that are released into the environment, because we have to account that we're going to have some fertilization that occurs. We also have to satiate those predators, because eggs are really nutritious. All of that yolk and those lipids that are laid down make a very good food source. And so we need to satiate all those predators and also accommodate for any stochasticity in the environment that might inhibit or that might make fertilization much more difficult. So imagine reproducing where you're just releasing all of your eggs and sperm into the environment in the ocean, and you're hoping for fertilization. Imagine you're on that nice sunny beach swimming with your significant other, and you get a mouthful of sperm and egg in your mouth. Most people are afraid of sharks in the ocean. I think I'd be afraid of fertilization happening in my nostrils. So this is something that happens in the ocean. There's a couple of varieties of internal fertilization, which is really the most fascinating type of reproduction for me. So is what's happening here. If you're not familiar with internal fertilization, I'm going to explain it to you. There's a female, and she has an internal reproductive tract. And there's a male, and he has sperm that he wants to introduce into that. And the way that he does that is with something we call a copulatory structure. So you might be familiar with the term penis. We're going to talk a lot about fish penises tonight. So this is your trigger warning, okay? If you're uncomfortable with penis talk, you can exit, and I will not feel bad, okay? So there's a couple of ways that this happens, and this is truly just the most fascinating part for me. Sperm is going to be delivered as close as possible into the internal reproductive structures of the female to increase the chances of fertilization of that egg. So in the case of something called an Ova Viviparous fish, an example is our rockfish here, and she's pregnant. I'm not sure if you can see this really well because of the light, but this is her pregnant ovary. Okay? So with an Ova Viviparous, oh, thank you. Now it's really romantic. So here, with our ovary, is what's happening is the male has inserted his sexy copulatory structure into the female reproductive tract. They have fertilized the eggs. And these fish have a lot of yolk in their eggs, even though it's internal fertilization. And the goal here is that the female provides protection for those millions of developing larvae. And when they get to an advanced state where they can feed on their own, she will give birth to them. So here, if you can see like the little kind of iridescent spots, these are the eyes of the fertilized eggs that are developing within the ovary. So when we have a situation like this in fish, we're familiar with the uterus in humans and other mammals, but in fish, it's all this embryonic development is occurring within the ovary. And so she's going to give birth to all of these larvae at some point in her, in the next few weeks after she is fertilized. Another form of internal fertilization is called viviparity. And in this situation, same thing is happening. Fertilization is internal, but in this case, the yolk content that would supply for the developing embryo is quite reduced. So the female is retaining these embryos that are developing and these young fish within her body cavity for a prolonged period of time. And the difference here is that she's actually providing nutrients and gas exchange to her developing young. So this is a female black perch that we've cut apart the outside of the body cavity, and you can see the developing fish within here. And if we dissect out the cavity here, this is actually the ovary, and there's multiple babies inside here. And here we can actually map out where each individual fish has its own sac that mom is contributing nutrients and gases through the blood circulatory system. Okay, so you might be thinking, why did they invite this woman? She's a little bit crazy. Well, like Julie said, I met a lot of check boxes when she was looking for a speaker. So that's how I got here, okay? I'm really interested in fish reproduction because it's kind of like a detective story to me. You might see a slimy, scaly fish, and you might think it's pretty or it has an interesting shape, but when I see a fish, all I can think about are gonads. How big are the testes? How big are the ovaries? And how are these fish doing it? That's what I'm thinking about. But it's actually more than that. There's a real dramatic how, what, and why to reproduction. How are they doing it? What are they doing it with, and why are they doing it in this way? So we can answer a lot of evolutionary questions based on structures, based on patterns, and we can actually take rare fishes that we know very little about and are able to describe a lot of reproduction based on some of the patterns that we've observed. And that's really my goal, is to kind of look at rare fishes and fishes that are important to us commercially and recreationally and look at their reproduction. So I'm going to share with you 4 stories of reproduction that are occurring in my lab. And the first one is on the oarfish. So in case you're not familiar with the oarfish, it's the world's longest bony fish. This thing is massive. It's pretty impressive, 17 to 20 feet long. Okay, so again, remember, this is kind of a crazy woman talking to us. She's thinking about gonads and how enormous these gonads are. Okay, huge fish. This is a deep water fish. They are found from about 30 meters to 300 meters and have been reported as deep as 1,000 meters. So it's a mid-water fish. So given that, we don't know a lot about it because we're not down there at that depth watching reproduction. So I'm really interested in oarfish. And it's amazing with a rare fish what we can actually find out with a few number of species, a few number of animals. So the oarfish, the only time we really get a chance to examine them is when they strand themselves on the beach. And this happens often during really strong El Nino and La Nina years. So in 2015, we had a strong El Nino, and we had 4 oarfish that washed ashore. So we had 2, one in June and one in September, 14 to 17 feet long and weighing over 200 pounds. This is a massive, impressive fish. So the first thing I want to do is get my greedy little hands on the ovaries. Okay. So in June, here is our oarfish, and these are my lab group that helped me. And this is the intestine and the stomach and the liver here. So you can see that this is quite massive. And here is the ovary stretched out on my lab floor, 7 feet long. My students were laying down, snuggling with it, taking selfies, weighing 24 pounds. Okay, so a massive ovary, the largest ovary I've ever been able to examine, and it was thrilling, I won't lie. I was giddy for days, okay? In September, we had another female wash ashore with a respectable 4.7 foot ovary that weighed a little bit under 3 pounds, so this is really interesting that we see this dichotomy. So as what we've been able to do, since no one's really described the morphology of the gonads of oarfish, is we took some pictures and we've kind of diagrammed how they fit in the oarfish body. So like other fishes, the gonads are located dorsally in the body cavity. So this represents the ovaries, this is the intestine, the digestive system, and there's the liver. Here is the ovary, the left and right ovary going down the length of the internal organs still attached. So is what's really fascinating about this enormous ovary is most fishes and most vertebrates have paired ovaries and paired testes. So there's 2 of them. They're typically of a similar size and they're located dorsally in the body cavity of a fish. In some fish species, we see that during development, one of the ovaries becomes vestigial and non-functional, and there's only one ovary that produces eggs for that fish. So I'm familiar with that. What we're not really familiar with is what we call a bifurcated ovary, which we identified in the oarfish. So the first third of this massive ovary, so the first third being closest to the head, there's two separate ovaries, two separate structures. The next third of this ovary, there's still two structures, but there's a very thin ovarian wall, unlike the ovarian wall that was around the two ovaries from the first third. And there's this incredibly thick connective tissue that holds it tightly together. This last third of the ovary is completely fused into one structure. So this is a very bizarre morphology of an ovary. And in fact, in that posterior portion of the ovary, if we do a cross-section and cut it in half, this diameter was about 13 to 14 inches in diameter and is filled with developing eggs. So I think that maybe this picture was one that Julie put on the flyer where I have my arms shoved up into it. Yeah, because that's what I do as a reproductive biologist. I want to see these big gonads, and then I want my hands not only on them, but in them. Okay? So it's okay for you to judge me. I understand. You'll have something to talk about at breakfast tomorrow. One of the things that we did in our lab is we took small chunks of this ovary, weighed it so we knew how much it weighed, and then I had my students, not me, count every single egg. So at first, and this was really fun because I had some high school interns in the lab and they were looking for something to do, and I'm like, I got a chore for you. And so I cut out these chunks of ovarian tissue and said, identify and count the different stages of eggs. And I let them to it. I told them how to identify them, how to count them. And we classified the different stages of eggs, so this is a piece of the tissue they examined, into primary, secondary, and tertiary eggs, or oocytes, that are going through these progressional developmental stages. Primary being the smallest, most underdeveloped oocytes that will progressively develop at some point in the female's lifetime. Secondary, they're kind of arrested, they're developing, but it's really our tertiary stage eggs that are going to be ovulating in the next breeding cycle or breeding event. So I left these students to it with a bunch of tissue and came back a couple of hours later, and they were up to 2,000 eggs in that little piece of tissue. I was like, you keep counting till it's done. And they did for days and days. All they did was count eggs. The purpose of this was to determine the fecundity of this female in these massive ovaries. So this is something we're really interested in if we're looking at fisheries biology, especially if it's something that we're capturing for food. What's the fecundity? How many total eggs can this female produce? And in this 7-foot-long, 24-pound ovary, she had approximately 86 million eggs. 3.6 million of them were tertiary oocytes, which means she was getting ready to spawn those. So I was really excited by this, slightly disappointed, because the longest fish in the world, I was hoping to be the most fecund animal. And I saw publications in Nature and Science. And then I learned about the mola mola, and that jerk has a higher fecundity. So we're actually ranked number 2. We're still above all the tuna, so I guess we have that. So we take a look at some of the histology. So again, those horrible microscope slides that you hated in high school. Here's our June fish and our September fish, and I've tried to outline some of the primary, the smallest ones, secondary, middle-sized ones, tertiary, larger ones. It's what we saw in the June oarfish. She had really big, healthy, maturing eggs, indicating that she was really close to a spawning event. In September, there were some large tertiary eggs that were developing, but none of them were healthy. And they're actually undergoing a process called apoptosis, which is a cellular death. This occurs after the female has already spawned, and there's always going to be a few eggs that are left behind that weren't quite ready to be spawned. And they're undergoing this cell death so that the female can reabsorb all of those nutrients that she was dedicating to those eggs. So this indicates that that September female had already spawned. So that's really cool with the female. What about the male? We were incredibly fortunate to have 2 males also wash ashore in 2015. Now, most of the oarfish strandings have been female, and there's only been a couple of counts of males. and nobody has ever examined the testis. So I was really excited in August of 2015 when we were told that we had a male oarfish. I had dreams of this fantastic testis, huge. I've worked with salmon testis, which can be 6-7 pounds, and I could not wait to get my hands on this huge testis. I came into the lab and almost cried. Here's the testis of this male. In August, it was 2 feet long and weighed 0.09 pounds. Just a few ounces. Tears. So I was excited in November we were getting another male because this is an incredibly unique opportunity to study these male fishes. And he had more impressive testis, 3.4 feet and a little over a pound. Okay, so in my lab, the motto is, size does matter. I don't know what you've been told. Size does matter. But the testis were still very interesting to study. And we did the same thing that we did with the female. We laid them out and we described them. Now, usually when you have an interesting ovary where it's bifurcated, we see a similar pattern in the male, in the male, so they're both would expect it to be bifurcated. This wasn't the case. The male still has 2 completely independent testes, and they were held together by this very thick connective tissue, the same type of tissue that we found in the female. One unique thing is that the right testis, it's just a little bit smaller than the left one. And that's okay, because they're still producing sperm, and it's still going to be enough to fertilize all of her eggs. So we did some histology on these males as well. So here's our August male. Here's our November male. I'll start with November because he's much more impressive at 2.4 feet. So I kind of describe the testis and these seminiferous tubules that I showed you before, which are the cross sections of where the sperm develop. It's kind of like a tree. So you can see these branches in both of these animals. And I kind of attribute that to a tree. So in our November fish, it looks like a spring tree. Lots of leaves, lots of beautiful, plentiful leaves, and you can hardly see the tree branches within there. There's so much sperm developing. So each one of these little purple dots is a developing sperm. In August, this kind of looks like a late fall tree. All the leaves have died and have fallen off, and you can see those branches. So in August, we didn't have really any healthy developing sperm, indicating that this August male already spawned, and our November male, he was really close to spawning, because when we brought this testis into our lab and cut it open, sperm spewed all over our lab floor. To a lot of people, this might seem disgusting. To us, it was really exciting. So for dead fish on a beach, what can we really know? Because we know that these patterns are consistent in fishes, we can kind of hypothesize about the reproduction of a deep water fish that's incredibly rare. So we also measure something called gonadosomatic index. So we weigh the gonad and we weigh the whole body of the animal, and we come up with this index. What percentage of the total body weight is composed of that gonad? And that tells us a lot. In June, our female was almost 12% gonad. And no, it seems like a small number, but that's really impressive. Imagine if 12% of your body was gonad. That's massive. Okay? And it had maturing eggs. So this animal was close to spawning and getting ready. Our September female, really low GSI, and remember, there weren't very many healthy maturing oocytes. So she has likely already spawned. With our males in August, really low GSI, not a lot of healthy maturing sperm. This male already spawned. Our male in November, a much larger GSI, mature sperm present, and it was in the process or getting ready to spawn. So that, in collaboration with sightings of larval oarfish off of Catalina, it really looks like the spawning for the oarfish occurs in early summer to early fall. So as what we think might be happening here is spawning occurred between June and August, and probably September, because this female has already spawned. And November, we had this ripe, mature male. So there could be another spawning event that was occurring, or we can remember that this was a very strong El Nino year where we had really warm waters. And warm temperature can increase the rate of gamete development, gametes being our egg and sperm. So it's hard to say for sure whether they go through another spawning event or whether this was an abnormality that we saw because of weather conditions. Is what we really need to complete our story here is to get more fish during the winter months and during non-El Nino years, which doesn't really happen very often. But everybody on this coast knows if there's an oarfish that washes ashore, Dr. Forsgren wants those gonads. So I'm going to change directions to another fish. This is the California Grunion, near and dear to Cabrillo's heart. If you're not familiar with the Grunion reproduction, I really encourage you to come to one of the public outreach event nights and come and watch fish sex on the beach. It's amazing. I really recommend it. So if you're not familiar with it, what happens is at the spring high tides between March and August, Grunion come onto the beach. The females burrow this little hole in the sand. The males wrap around the female and release sperm. Magically, those eggs get fertilized, and 2 weeks later at the next spring high tide, those waves are going to bring those eggs up to hatching and wash them back out to shore. So when I arrived at Cal State Fullerton, Kathy Dixon, another professor there, was studying egg development and how the embryos are hatching at different rates based on different temperature, thinking about climate change and how maybe sand temperature could be different and how this could alter larval development. And one of my first days on campus, she came by and said, "Hey, so I think that these Grunion have a penis." And I was like, "Cool, let me see it." Because that's the kind of person I am. So what they discovered was here on the Grunion, there's a little papilla. And this is what it looks like. It's just this little innocent looking structure on the underside of the belly of the fish near the anal fin. We never saw it in the females. So what Kathy Dixon would do is go out and collect the Grunion, and if you apply just the lightest amount of pressure to the abdomen, eggs and sperm come out, and you can fertilize your own eggs. So we saw this present on the male, but not on the female. Here's a video of sperm coming out. So you might want to turn the light down just a little bit so they can see this. So we're applying pressure here to the abdomen, and this video will repeat this. You can see this clear structure is the papilla, and sperm is coming out. So we're pretty convinced that this is a structure, a copulatory structure in this fish. Now, our copulatory structures, remember I said that's our way of getting sperm into the female reproductive tract for internal fertilization. And here is an externally fertilizing fish that has a penis. So cool and very interesting, right up my alley. So we looked at the histology of this, again, taking our papilla and doing a cross-section and looking at it. We can see the digestive system, we can see a urinary opening, and we can see a genital opening. And this is where the sperm is coming out. And we know that the sperm is coming out because we saw from the video, but also our histology, we see large numbers of mature sperm within that cavity. So just for comparison purposes, we wanted to compare this to the female. Again, when we apply pressure to the abdomen, we don't see this papilla that's emerging from the female, just eggs coming out. And again, with the histology, the digestive system is off of the slide, but here we have the urinary opening, and it opens up directly to the ovary. So there's not really a papilla structure. She's just going to release the eggs directly out of an opening from the genitals. So a technique that we've started using in the lab is looking at magnetic resonance imaging, or MRIs. And so we take MRIs of the fish, and this gives us a little bit more information about the content of the abdominal cavity and the gonads within that. So here's an MRI of a female, and you can see the eggs, and you can see the anus, and the urinary tract is really small. You can't really see it here in the MRI. And here is the male. And what we did with the male is we started color coding all of the different structures here in this MRI. The green is the urinary tract, the blue is the testis, and that pink color is muscle. So it's what we've done with the MRI on the males. Here's some histology, again, showing digestive system, urinary tract, and then we have the testis coming out. This is the same viewpoint here with the papilla in the male. So we've decided that this is a urogenital papilla. So there's an opening for the urinary products and also for the testis products. And the green line here, again, is our urinary system. The blue is the testis, and this pink is skeletal muscle. And as what we're able to do is render these three-dimensional models so that we can look at a testis and a copulatory structure in a three-dimensional way, which is really exciting if you're into this kind of thing. So we've decided that they have this urogenital structure. So why? This is part of our detective story. Remember, our female is burrowing down into the sand to lay her eggs, and the males are surrounding on top of the beach and releasing sperm. So if he didn't have a specialized structure, sperm is going to spew onto the beach and be wasted on the sand. So the amount of skeletal muscle and the attachment of that skeletal muscle to the body wall that we see here gives him a lot of control to aim that papilla down into the female's hole in the sand. Additionally, that pressure from contracting the muscle is going to apply pressure onto the testis and project the sperm all the way down into the hole, into the female's nest, fertilizing those eggs. It's also a really great sexual dimorphic trait that we can determine males and females without invasively looking at them. So that's a really neat tool. So another favorite project of mine in our lab is looking at the black perch. And this is a fish that's also really commercially and recreationally fished for and is important along our coast. And there's a lot of information out there about some of the perch species, particularly the shiner perch. And they've described reproduction, and they've described how the sperm enter into the ovary, and that the female can hang onto the sperm for a prolonged period of time before she fertilizes her eggs. So a really interesting mode of reproduction where you have a fish that can decouple copulation from fertilization. But this is a perfect story of how you can't take one species in a family and apply it to every fish in that family. So we take advantage of how one fish reproduces, but it cannot be applied to others necessarily. So as what we were interested in my lab was, how are they making this sperm packet that gets transferred to the female and can be stored for long periods of time? Because a black perch mate in early summer, hang on to the sperm for several months, fertilize in the fall, and give birth in the spring. So what we've done is take a look at the testis, and again, here's some more histology pictures. The seminiferous tubules where all of the different sperm are developing are here. So the lighter brown, reddish colored are less developed sperm. And you'll see we have an enlarged picture of this little cavity, and these are mature sperm. So the sperm head is this dark purple dot, and kind of this brownish color are their tails. And so what we believe is happening is within the seminiferous tubule, all the sperm are developing at the same rate. And so they're all going to be mature at the same period of time. And they're grouping together, they're clumping together in that seminiferous tubule before they're released from the testis and externally. And then what's happening is once these sperm are coalesced together in a group, they further combine into the spermatophore, which is just a great big cluster of all these little tiny clusters of sperm, and that entire spermatophore is going to be transferred into the female so that she can hang on to it and decide at what point she wants to fertilize her eggs. So if I've taught you anything yet, you're wondering, what does that look like? What is that structure that's transferring that spermatophore? So here it is. Here's our male black perch, and on the anal fin, we have what's called a flask organ, and this is a little protective structure, and inside of it is the intromittent organ, and you can see the tip of it here. Now, the fish has two of these, one on each side of the anal fin, and here's a little video showing if you apply pressure, oh, hello, there it is. That's our intromittent structure. This is the copulatory structure. So the male is taking that spermatophore, he's going to insert this copulatory structure into the female's reproductive tract and introduce that spermatophore. So we're really interested in these intromittent organs. So when we get small fish, so young of the year, and the black perch, if you remember from earlier, is one of the fish that's going to retain the eggs. The babies develop within the ovarian cavity, and she gives birth to fairly large, mature fish. So when we see these young of the year, there's not an intermittent organ. These fish are not sexually mature yet, so they don't have the intermittent organ. They don't have the ability to copulate yet. But when we look at the histology, we actually see this red here are the fin rays that correspond with this here. And this little patch, we can actually see specialized tissue that will develop into the intermittent organ. So, and a lot of mature males that we see outside of the breeding season also don't have this intermittent organ. So we think that in some animals this may be regressing, but we haven't caught enough animals to really take a look at this. So here's a really good ALTA-C project. I want to collect a bunch of these juveniles, expose them to testosterone, because I think that's what's inducing it, and see if we can make intermittent organs. So let's open that up soon. So what we've done with our histology is we've really pieced together this spermatophore and how it's being transferred. Here we've put together the testis and our collecting duct, and this is the direction in which those spermatophore are being transferred. They're going through the body and they're exiting this intermittent organ. So here's what the genitalia structure, that intermittent organ is placed here on the anal fin, and this is what the testis look like, and that's what we're kind of theorizing is happening with this sperm transfer. So my graduate student, Evelyn, has been working on this project. And she spent the summer in Friday Harbor, and she looked at a lot of surf perch. And somehow she's inherited this innate love for fish penises like I have. And every fish that she's been able to get her hands on, she's looked at them. And it's really interesting what we've noticed. These closely related species have different types of structures. So here's our black perch with the intermittent organ. Again, there's two of them on that anal fin. When we look at our shiner perch, they have an intermittent organ again on the anal fin. They have a fleshy papilla here too. And we see this in the walleye surf perch. Again, we have a pair of intermittent organs and a really teeny tiny, but it's there, papilla. So why do some of these fish have 2 copulatory structures and some of them have 3? That's our detective story. We're really interested in figuring out, does that testis go both to the intermittent organ and to the papilla? And what are they doing with this? And I'm also interested in what does that female genitalia look like? It can accept an intermittent organ and a papilla. This has to be pretty interesting as well. So the last project that I'll talk to you about tonight is the blue rockfish. Again, really important commercial and recreational fishery on our coastline. And a lot of this work is being conducted by my graduate student, Hamam. So we're looking at the urinary bladder. Okay, now you're thinking, she's really crazy. Okay, here's some dissected fish. The testis kind of runs along here dorsally. This is all gut. And this is the urinary bladder of a male. Over here, the female, we have the ovary, stomach, some intestine, and the urinary bladder is this little tiny structure. And the male urinary bladder is longer and bigger and weighs a lot more than the female. And this is occurring in all different species of rockfish that we've examined at all different time points in the year. Okay, why? This is pretty bizarre, right? Well, when we start looking at the mating behavior of rockfish, it's been described several times, the courtship of what's happening, where you have pairs of rockfish that are coming together and the male is swimming about the female. So here's a video of some Japanese rockfish, and this is a female. She kind of cues and stays stationary, and it's the male who swims about her. And if you watch his tail, it looks like he almost slaps her in the face. What we think he's doing is urinating in her face. Okay? So this was something that Dr. Milton Love from UC Santa Barbara, if anybody knows him, he's a little bit crazier than me. He pulled me aside one day and said, hey, do you think that rockfish could be peeing in female faces and have a pheromone? And I was like, oh yeah. Yeah, they can totally do this. Let's start looking at this. So I waited on this project till the perfect student who was interested in some weird stuff, came along, and we're looking at the urinary bladders of rockfish. So as what we've seen, here's some more histology. Here's our female and our male. So the urinary bladder is structured similarly to other vertebrates, where it's covered with an epithelial tissue on the outside. Okay, so here's your bladder, and on that inside cavity are epithelial cells. And then surrounding that is smooth muscle. That's what's allowing you to hold your urine right now. Okay, it's also what's helping you to control to release it. So when we look at the males, and these are taken at the same magnification, we have the epithelial tissue lining here and smooth muscle here, but as what you'll see are these secretory structures. And these are found throughout the entire complete urinary bladder. So there's been some work done on pheromones produced in urine in freshwater fishes, and they've looked at these secretory cells and confirmed that they're producing a pheromone, and that's what we think that we have here. We think that our rockfish are producing a specific pheromone, and then during this courtship, the males are presenting that to the female, which means a female must have receptors in the olfactory tissue picking up on that, and that will give her a cue as to the quality of that mate. Nobody's ever observed the actual copulation of these fish, but we have observed this mating behavior. Because after the female selects a male, they go privately in the cave and do their business. So again, we're really interested in their copulatory structures, and they have a papilla as well. So when we find something, we want to know how it gets transferred. We're looking at the urogenital papilla of the rockfish as well. Because when Hamam started going on fishing trips and collecting rockfish, he would tell me, the fishermen can sex these things by the papilla. And I was like, what? That you can tell the species by their genital papilla, which is really interesting. And they have different shapes. So this is our blue rockfish, and this is the genital opening and the urinary opening. And that urinary opening is much larger than the genital opening, and that's probably to accommodate a large amount of urine that is being directed towards that female. So here's a close-up picture of the genital opening and the urinary opening, and we've actually had them spewing sperm right in our hands, which is pretty exciting if you're into that thing again. Histology-wise, we're able to identify the urinary bladder, and that genital opening inside of that is mature sperm. So we know that that's how that they're transferring both the urine product and the testis product. So that copulatory structure, again, we're really interested in what's happening into the female. And when we examine the female, we see the urinary opening and the genital opening for the female, but she has some pretty impressive genitalia. So we're starting to look at the female genitalia and how she's able to accommodate the copulatory structure of the male. And again, here you can see kind of the papilla, and behind that is the opening of her genitals. So here's the opening, which is where theoretically that papilla, where the male would insert and inject the sperm, and here is the urinary opening. So we've got some undergraduates in our lab, and their task is to specifically look at the genitals of these different rockfishes. And so as what we found, like I mentioned before, the shapes of these are very unique and different for every species. So in some, they're quite impressive, like our copper rockfish, but the vermilion, we can see the urinary opening, but the genital opening is really quite small, and that's very similar to what we see with the starry rockfish. So the question here is, do they have a copulatory structure that's similar to the Grunion, where they're able to hold it in internally and then they just bring it out at the time of copulation? Or is there really not much of a structure there, which means that the male genitalia must come very close in contact with the female in order to accurately and effectively transfer the sperm? And then we have some really big, impressive genital openings, like in our speckled rockfish. And there's the papilla, and there's the urinary opening. So what do copulatory structures, testes, and ovaries have to do with you? It's a very fair question to ask. And so what I'm here to tell you is that whenever you take a bite of that fish, you should be thinking about how it got there. How is this fish made? What do its testes look like? What do its ovaries look like? And how does it relate to you? Well, the simple answer to that, besides we're all fish porn enthusiasts now, is our babies need fish babies, and they do. The fish provide a huge amount of protein to the world's population. So in order to keep providing a protein source to our babies, we need fish babies, which means that we need to come up with effective management strategies in order to manage the populations that we fish for. So just to give you an example of what we do with fish, we love to fish, we love to eat them, we do it recreationally. So here's my niece, this is a freshwater fish, don't judge me, that's cool. But we eat a lot of it too. And fishing is a huge tourism event. So a lot of the economy is based not only on trading fish products, but also in attracting tourism and recreational fishing. So we need to think about all of these different aspects and how they contribute to our economy and how they contribute to our food sources when we think about managing fishes. So in California in 2015, we harvested over 96 million fishes commercially, and over 5.5 million fishes recreationally. That's a lot of fish for our 38.8 million residents in California in 2015. So we fish for a lot of fish in California. We have a lot of residents that we're feeding. How do we look at this on a global scale? Our world population is about 7.5 billion people right now. And in the 1950s, we started tracking how much fish was removed from the ocean worldwide. So in 1950, we were looking about 20 million metric tons that were removed from the ocean, feeding a population of 2.6 billion. So if we project our population in 2043 being 9 billion people, what do our oceans and our fisheries look like? So in the late 80s, aquaculture became really popular. So this blue is a trajectory of how aquaculture production has increased over the last many years, several decades, and how our maritime collection of fish has also increased up through 2014. So in 2014, we removed over 90 million metric tons of fish from our ocean. So reproduction is really important in how we manage our fisheries. A lot of fisheries management, so I know if there's some avid fishermen out here, you might be angry when the Rockfish fishery is closed, because we're trying to protect the species so that it can properly reproduce and give you another generation of fish to catch the next year and the next year and the next year. A lot of our trajectories and a lot of our predictions are based on that one species that we know about, and we're projecting that on an entire group of fishes. And that doesn't work for all species. So we've been collecting rockfish and looking at papilla and urinary bladders for about a year now. And our rockfish fishery is closed from about January to early April to allow fish to breed. But that big, ripe, blue rockfish ovary that I showed you at the beginning was collected in November. So we're not adequately protecting all of the species all of the time. So we need to come up with really good reproductive understanding so that we can come up with better management strategies, not only for how we're going to manage our freshwater and marine fishes that we're taking, but also for aquaculture. So aquaculture feeds a large amount of people worldwide, and reproduction is directly applicable to aquaculture because we need to know what are the stages of ovarian and testis development, when are they sexually mature, how do we fertilize eggs, and how do we get the biggest, best growing fish that we can put on the plate. So these are really big concerns that we have coming up in our future with the number of people that we have on our planet and the pressures that we're putting on our ocean. So humans have a huge impact on our oceans, and we know that. You guys know that. You're here because you're interested in fish and in our oceans and how we protect them. But for the majority of people, they never see the ocean, and they don't understand fish, let alone fish reproduction. So we have to be able to communicate to people in our communities that we have this invisible environment, the ocean, that has fish, which is a huge amount of protein that we consume on a regular basis in order to sustain our own lives. So these are a variety of things that are affecting our fish population, and everything that I've highlighted in yellow is directly affecting reproduction, but is very, very little studied. So I've also been collaborating with Orange County Sanitation District, and I've been doing some work looking at pesticide effects on ovarian development. Super data heavy, kind of boring, so I didn't share that here with you today, but I'm happy to talk to you about it. But we definitely see that some of the pharmaceuticals that are making it into our waterways, some of the urban runoff, some of the pesticides that we're utilizing, plastic compounds are estrogenic, petroleum products are estrogenic and act like a testosterone. These are all things that are going to affect fish reproduction. And in order to fully understand those impacts of our fish populations, we really need to have a good understanding about how fish reproduce, why they're reproducing, and what they're using to reproduce. So with that, I'd like to thank all of the students in my lab who painstakingly dissect gonads and count eggs, and everybody who's really influenced me and gotten me here to this point. And with that, if you have any fish porn questions, I'm happy to entertain those.
Well, Christy, I think we have some time for some questions.
Yeah, excellent.
Just what you had hoped for.
I mean, I love talking about fish penises even more, so bring it on. Yeah.
I found it very interesting on was it the oarfish, the large ones that were washed up, that in both the male and the female, the larger ones seem to have the smaller or less healthy reproductive organs. And I was wondering, do you have a way of judging age on a fish, and could that be age-related?
Well, I mean, you can definitely look at age. We do that with otoliths, the ear bones. But by externally looking at a fish, I can't really, it's not like going to the grocery store and I'm like, she's definitely over 60. That one's definitely 20. So we don't really have a good way of externally judging them. But I hope I didn't give you the wrong idea that they weren't unhealthy. They're perfectly healthy, other than they were dead on a beach. There's a huge difference in the size. That's because they've previously spawned. So if you think about a fish that's going to mature gametes, those eggs are pretty large relative to a sperm. So eggs are really big. There's a lot of yolky materials within them. And the female invests a lot of energy to that. So they get big. And as she gets more eggs that are developing, they're bigger and bigger and bigger, and they weigh more. When she has a spawning event and releases them, they're pretty small again.
Well, that's why I was wondering if the bigger fish were maybe older fish.
They might be. Yeah, it's very possible. And we see that with a lot of fishes, especially our sex-changing fishes like our California sheephead. This is a species that's female first, changes sex to males, and it's a reproductive strategy that the bigger male is able to hold a harem and protect them and has pretty big testes. Yeah, whereas a smaller fish would have relatively small testes, not be able to produce the sperm to fertilize all the eggs that a female would produce. So definitely we tend to attribute larger fish with larger gonads and producing more sperm. But as far as the oarfish, they were all pretty much about the same size. You know, lengthwise, weight-wise, about 178 to 206 pounds. They were all sexually mature. They all had the ability to produce sperm. And one related question I get is, why the big ovaries and why the little testes? I don't know. Obviously, the males are still producing enough sperm in those little testes to fertilize the eggs. But if you think about the midwater environment where these fishes live, it's deep, it's dark, there's not a lot of fishes. Finding a mate might be difficult. So one thing that I've theorized is that maybe the females migrate and will spawn with multiple males in a particular area, and the males stay stationary. So they have the ability to have a small testis and still fertilize the females that might happen to come around. So rather than have both males and females going in this huge, vast environment trying to find one another, males hang around and sit and wait for females to come. And we see this in a lot of bird populations and other mammal populations where males remain stationary, have smaller testes, and it's the adult females that come to them. So really good question, yeah. Big, huge fish, super impressive gonads. Yeah.
This is like a two-part question, sorry everybody. With anglerfish, have you ever observed multiple males attaching to a single female? And also, can you go through sperm delivery from the male?
Oh, so in the anglerfish, okay, this is the one that has the parasitic males. So the females have parasitic males. And there's an anglerfish on display at Cabrillo here, and there's two males that are attached. So is what's going to happen, again, we're in a deep sea environment where finding a mate is really difficult. So these guys have solved that problem in a very unique way. The males, when they're small larvae, smell out the female, and she must smell really good. When they find them, they bite onto them. And what happens is essentially all of the male's organs, except for the testis, are going to be consumed by the female. And it's now a parasite because it's now gotten into the blood circulatory system of that female, and she's feeding him. But her ulterior motive is she's now got her mates attached to her, and when her ovaries are ready and she's ready to fertilize, ba-boom, she's got males who are ready. We believe that there's probably hormonal cues that the female is producing that acts on the testis within that little rudimentary male that allows him to mature the sperm. The actual sperm transport from the male to the female, we don't know. There's only a few anglerfish. It's really hard when you have a rare species that they want to let me cut out the gonads. Yeah, in the back.
You obviously get pleasure studying fish sex, but have you determined that the fish get pleasure having sex?
Okay, good question. I was hoping somebody would ask me about fish orgasm. Yes, absolutely. There's actually a book written on fish orgasms, okay? So the whole process of an orgasm, if you just want to get down to the real basics of it, is just a lot of muscular contraction. And that muscular contraction is usually on the female's part, and that muscular contraction is to help give that little sperm a little extra oomph in that reproductive system to kind of get it to where it needs to go. Never really been described in seawater fishes, but there's several freshwater fishes. Your guppies at home, any of you have guppies at home? They are sexual deviants. They have orgasms. And what we think too is that if there is a male that's present in the laboratory, it's been observed that the females will undergo an orgasm, fake that they've previously mated with a male so that male doesn't come by. So yes. Good times, right? Yeah.
You mentioned that you're a professor at Cal State Fullerton. Do you teach like a fish reproduction class?
I'm teaching a reproductive biology class right now. So yeah, at Cal State Fullerton, I primarily teach physiology. I teach a lot of human physiology. I teach comparative animal physiology. Reproduction is always part of it because we need to know a lot about reproduction, whether it's fish or humans or whatever. In the comparative class, we talk about how agricultural animals reproduce because that's really applicable for students interested in veterinary school. And this semester, I finally convinced our department that people are interested in reproductive biology, and I'm teaching a reproductive class. I have some of my students here tonight. It's not specifically fish reproduction because I was kind of worried that I wouldn't get a lot of students just for fish. We cover everything. Duck penises with lassos, monitoring penises. They have 2, all kinds of stuff. Weird vaginas, 3 vaginas. So, yeah. Come visit me if you want to have a whole semester long of this crazy talk. Yeah.
I was wondering if you could describe how climate change affects fish reproduction.
Yeah, so when we talk about climate change, there's a lot of different aspects to that. When we think about our oceans, we typically think about how the water may be increasing in temperature. So a lot of fishes reproduce based on temperature cues. So if we take a perfect, healthy environment that we're not concerned about, we want to produce our babies when it's springtime and there's lots of food resources available for them. So in the spring, the sun is out longer, the water temperature is a little bit warmer, the phytoplankton are starting to bloom, they're producing food for other fishes, and there's a lot of food available for larger fishes. So spring and summer is a really good time to reproduce, and that's on our coast, not necessarily tropical areas, or the Arctic and Antarctic. So with climate change, what we can see is that water temperatures are increasing. So that water temperature is going to cue fish to reproduce at an earlier time. This has huge implications for an animal that's depending upon temperature cues to breed. So if the water temperature has increased, and we don't necessarily have a longer photo period to increase our phytoplankton, we now have the potential to birth babies when we don't have food available. So that's one aspect. We also have salinity changes, pH changes. It's been shown that changes in pH disrupt the egg membrane. So it can disassociate that, making fertilization harder, or completely destroy the egg so that there is never going to be fertilized anyway. So there's several different aspects that climate change can influence reproduction. Temperature is the most obvious one. Very difficult to study, though.
Maybe
Yeah.
one more question.
Oh, sure. I saw a hand somewhere. Yeah.
You mentioned the impact of plastics in the ocean.
Yep.
With ultraviolet rays breaking down the plastic and the chemicals released, it's theorized, and there's some proof that there becomes a hormone disruption.
Yep.
I'm wondering if you have observed that, it's evidence in any of the
I
tests.
haven't really looked at that per se. It's really hard to go out there and say, hey, this fish was exposed to plastics, what's it doing? In the laboratory, there have been experiments with different types of plastics and the compounds they're producing. And we do see that they do act like estrogens. And estrogen is a really important hormone that in those tertiary eggs that I showed you, estrogen is what's promoting that development. And so is what happens when you have an exogenous or external hormone that's acting on the reproductive system. My famous example is, think of a bodybuilder, right? These big, huge, buff guys that are taking anabolic steroids to build muscles, okay? So they're taking an exogenous hormone and introducing it into their body. Is what that does is it feeds back to the hypothalamus and the pituitary, which is regulating the gonad. Now, the funny thing that I find about these big macho guys with these huge muscles, their testes are about the size of a pea because they've shut down testis production of testosterone. They're no longer producing sperm because they're taking that testosterone exogenously, shutting down the hypothalamus and pituitary. So in the case of plastics and other types of compounds, pharmaceuticals are estrogenic. We also know that there's some that are androgenic that act like testosterone. This is happening in urban runoff, so petroleum products are estrogenic. Pesticides, which I've done some research on bifenthrin, they're estrogenic. So now you have a fish that's exposed to an environment where we can have levels of estrogen that have the potential to shut down that brain regulation of the gonad. And it has huge consequences, or it can. We can have situations where it might advance ovarian development, so now you have a fish that might be exposed and have the mature eggs, but the male has never received its testosterone cue because it's maybe breeding seasonally and there's no mature sperm to fertilize those. So by the time the male is ready, those eggs have probably died or been laid without being fertilized. That's one problem. Another problem that I saw with my bifenthrin experiments when I was looking at pesticides, and I did this with zebrafish, sticklebacks, and steelhead up from Northern California, and is what I see with them is that these estrogenic-like compounds are actually causing that cell death of the egg. So it's causing them not to really develop, it's destroying them, and they're not able to produce as many eggs, so fecundity is going down. Interestingly enough, males are not affected by it. The males, no problem. Testes were always perfectly healthy, but the females were not. And so that's going to cause a problem when we have animals that are exposed to a potential compound that can reduce fecundity. You're not going to be producing a large number of animals. And steelhead is a salmon that we really care about in California. California, we want our salmon. Our salmon are in these agricultural areas that are heavily impacted by runoff, particularly from pesticides. So we don't really have large salmon populations because we've dammed and we've done a lot of things to the waterways, but we're also introducing compounds that can act like hormones. Those are called endocrine disrupting compounds. Yeah, we see that a lot.
Well, Dr. Forsgren, I'm sure I'm not the only one that is now thinking a lot about reproduction.
Excellent.
Fish reproduction.
And it's a Friday night, it's still kind of early.
So thank you so much.
Thank you for
being
inviting
here
me.
tonight.
Thank you. Thank you so much.